afternoon, everyone. My name is Haylee D'Agui. I'm a Research Associate at the ARC Centre for Mine Site Restoration at Curtin University. Today I'm presenting on functional and biological analysis of topsoil at mine sites in Western Australia. I undertook with co-authors Justin Valliere, Mika Vanderhyde, Paul Neville, Ben Moreira-Grez, Marsa Musavi Duraz-Mahale, and Kingsley Dixon. Thanks for tuning in. You would have just heard from Justin Valliere on his part of the study, and I'll be expanding on some other angles of the same study. Native topsoil is a scarce, highly valuable resource in post-mining restoration. It is high in organic matter and nutrients and contains plant propagules and microbes. Topsoil is often stockpiled during the mining process for use in restoration post-mining. A variety of factors such as the size and depth of stockpiles, duration of storage, climate and soil type affect the functionality of the topsoil when it is eventually used in restoration. The mining industry often has insufficient understanding or inadequate tools to assess the impact of storage on the vitality of topsoil as a restoration resource. A key component of topsoil health is ensuring a competent microbial community. However, soil functionality in restored soils has been found to be non-comparable to that of native undisturbed soils. Here we aim to determine how the diversity, abundance and activity of the soil microbiome of stockpiled topsoils is influenced by factors such as storage time for different regions and commodity types in the major mining provinces of Western Australia, with the main study objectives being to determine whether soil microbial communities, soil respiration, selected plant growth parameters such as biomass, number of root nodules, and selected soil chemistry variables differ between reference and stockpile topsoils. To determine whether soil microbial communities, soil respiration, selected plant growth parameters, and selected soil chemistry variables newly established stockpiles are more like reference sites than older stockpiles. To determine whether there is a simple rapid assessment surrogate for plant growth, to determine whether there is a pattern across regions, and of course to contribute to the development of science-based topsoil management guidelines in order to optimise biodiversity recovery in mining soils for improved restoration outcomes. These are just preliminary results, and we've got a bit more work to do before we have it all finalised. We sampled topsoil from stockpiles and native reference sites at 7 mine sites across Western Australia. The BHP mine site in the Pilbara, the Tronox Cooldooroo operation, Alcoa's Huntley mine site, Fortescue Metal Group's Chichester operations, a mine in the Goldfields, Independence Group's Nova mine site, and a mine in the southwest of Western Australia that has chosen to remain anonymous. Our study sites included iron ore, nickel, bauxite and mineral sands mines and represented the Mediterranean shrubland, arid zone savanna, semi-arid woodland intersown and forest biomes of Western Australia. At each of the 7 mine sites, we sampled 3 reference sites and 4 stockpiles. At each of the reference and stockpile sampling sites, we established 5 5-metre by 5-metre quadrats and used a trowel, auger or pickaxe to sample soil from the 0 to 20 centimetre soil profile. We collected 5 replicate samples from within each 5 by 5 metre quadrat using sterile methods and bulked these samples to create 1 composite sample per quadrat for a total of 5 soil samples per stockpile or reference site. As such, we assessed a total of 35 soil samples per mine site and 210 samples across all 7 mine sites. We used a combination of methods to assess soil functionality. We recorded site characteristics including stockpile height, stockpile type and amount of vegetation present, and soil compaction. We assessed soil chemical and physical properties, including bulk density, pH, organic carbon, nitrate nitrogen, iron and other soil nutrients. We used 1-day CO2 tests to assess microbial respiration, which can be used to infer microbial activity, and we used next generation sequencing to determine fungal and bacterial composition. As per Justin Valliere's talk just prior to mine, we also conducted a plant growth assay to test the growth of the study species, Acacia silicona, in soil from each site. The ultimate measure of the health of the soil is how well it can support plant growth. As per the previous presentation by Justin Valliere, we conducted a plant bioassay to determine whether differences in plant growth were exhibited by plants grown in reference or stockpiled soils. We saw significant differences in plant performance. Overall, plants performed better in native reference soils than in stockpiled soils in relation to total biomass, root nodulation, and water use efficiency. As can be seen here in these figures courtesy of Justin Valliere, stockpiling generally had a negative impact on biomass and root nodulation. All of this information produced from the plant bioassay is useful and provides insight into soil functionality, However, we were hoping to be able to find some soil characteristics to measure in place of performing a time-consuming bioassay. This brings me to our chemical, physical, and microbial measurements. There was a clear separation between reference and stockpiled soils for mines F and G. However, as presented in this PCA, for most sites, the abiotic properties and carbon dioxide production show no clear differences between reference soils and stockpiled soils. So abiotic properties alone did not give a good indication of how seedlings would fare in a plant bioassay. When biotic properties, namely soil microbial composition, were assessed, we found minor differences in bacterial and fungal diversity overall. Bacterial diversity was highly variable within sites. Half of the stockpiles had higher fungal diversity than reference sites, and the same is true for bacterial diversity. A wide range of bacterial classes were represented in soil samples, and abundance of the different classes varied across mine sites, as well as between stockpiled and reference soils. There was also a range of fungal classes represented in the samples, and abundance of the different classes varied across mine sites, as well as between stockpiled and reference soils. Of note is the high abundance of fungi present in the mine F reference soils. As I mentioned at the beginning of this talk, these are preliminary results, and further exploration of the data is yet to be undertaken. Some clear differences existed in plant performance between plants grown in native reference soils and stockpiled soils. However, while it would be nice to be able to provide a clear recommendation of simple soil variables that could be measured to predict this, soil biotic and abiotic properties interact in a complex way. The challenge remains to untangle this and identify simple soil characteristics that can provide indications of the ability of the soil to support plant growth. Previous studies have indicated that microbial health can be used as a surrogate for soil functionality. However, assessments of microbial health in this study did not affect plant performance. This study assessed the 0 to 20 centimetre soil profile of topsoil stockpiles. It is possible that radically different results would be obtained if soils from deeper within the stockpiles were assessed, which begs the question, does topsoil stored deep within a stockpile exhibit the same functionality as the soil stockpiled on the surface of the stockpile? Another point of investigation could be the effect that revegetating stockpiles with native vegetation and therefore stimulating biological activity has on microbial composition and physiochemistry of the soils. Thank you to the ARC Centre for Mine Site Restoration, Curtin University, Independence Group, Fortescue Metals Group, Alcoa, Tronox, and BHP for supporting this study. Thanks also to the field and lab assistants and site staff that made the study possible. A big thank you to Dr. Justin Valliere for his collaboration on this study. Please feel free to reach out to the team with any questions. My email address is on screen. Thank you for listening.